it's not possible to eliminate all self-reactive T cells. It's not possible in the thymus. So because we do not have all self-antigens in the thymus, so we need some other tolerance mechanisms in order to prevent the activation of. Please, Hajiye Khanum. Please. Khanum means lady in Persian. For example, we say Agha for men and Khanum for women. It means lady and gentleman, something like that. So Hajiye Khanum means lady Hajiye.
Lady who wants to make. Lady who wants to make.
I didn't understand. So, I was talking about peripheral tolerance of T cell. As I'm telling you, we need some, you know the reason now. You know the reason why we need peripheral tolerance. Am I correct or not? So, the mechanisms of peripheral tolerance. The first mechanism, there are several mechanisms that prevent the activation of self-reactive T cell in the peripheral tissue and secondary lymphoid organs. One of these mechanisms, this is a normal recognition and activation of the T cell. We have an APC presenting antigen in the MHC and peptide TCR first signal and B7 CD28 co-stimulators and second signal. Then we will have T cell activation and effector and memory cell production. This is a normal process of T cell activation. It's familiar with you, yeah? So, sometimes we have three different mechanisms, including energy, suppression, and deletion, that I will talk about them one by one in more details in the next slides. We have three different mechanisms by which the peripheral tolerance prevent the activation of self-reactive T cell. So, as you saw in the previous slide, we have co-stimulation, signaling, the other co-stimulation. This is a normal response, and proliferation of the T cells, effector cell, and memory cell production. But sometimes, and this cell is a naive cell, and you know that naive cells need co-stimulation. Without co-stimulation, we won't have activation of naive cells. Okay? Okay? Without co-stimulation, we won't have activation of naive T cells. So, here, when we have recognition of a self-antigen, this is a self-antigen with naive T cells in the absence of co-stimulation. In the absence of co-stimulation. When there is no co-stimulation, the cells will be suppressed. A state of non-responsiveness will induce in the cell, which is called anergy. Anergy means unresponsiveness. Okay? As a result of the absence of co-stimulation. When there is no engagement of co-stimulation. Is that clear? When there is no co-stimulation, we won't have activation of naive T cells. Not only we will not have activation of these naive T cells, it will go to an state which is called anergic or unresponsiveness state, and we won't have any immune response, any activation here. Clear or not? So here the point is co-stimulation or co-stimulators. There is no B7 here. When there is no B7, there is no second signal. When there is no second signal, there is no immune response. It is called anergy. Okay? Clear or not? Let me ask a question. This is a self-antigen, but this is an APC. This is an APC. And this is self-antigen. but there is no co-stimulation. Why we have self-antigen, but there is no co-stimulation? What if an APC phagocytose A, for example, pathogenic bacteria, it will have co-stimulation? What the difference between self-antigen and, for example, an Escherichia coli antigen, that in Escherichia coli, APC will express B7, but in self-antigen, it won't express B7. What's the difference between self-antigen and E. coli antigens?
These cells recognize antigen in the thymus, but this don't recognize antigen in the thymus.
Do you mean T cell?
Yes.
No. We have two antigens. We have two different antigens. First antigen is a self-antigen, and we have a T cell that is reactive to it, but there is no co-stimulation on the APC, so there is no immune response. Okay, is that clear? Please have a look here. Is that clear?
Yeah.
So, but we have so for the first antigen, which is self-antigen, there is no co-stimulation. But if the antigen, for example, is an E. coli antigen, the APC will have co-stimulation. What's the difference between self-antigen and E. coli antigen? That E. coli antigen can induce co-stimulatory expression on the APC, but self-antigen cannot induce co-stimulators on the. Yes, I know the nature of them is different. Of course they do. What is the difference? No, they can be degraded to the peptide. All of them have peptide. The peptide is self-peptide. Sometimes, for example, we have an MHC, this E. coli peptide, but there is co-stimulation. The difference is co-stimulation.
Yes, because in E. coli antigen, there is the inflammation, some cytokines, so this suppress some of co-stimulation.
Yes, when there is E. coli, we will have inflammation, we will have engagement of PRRs by recognition of the PAMs. So there is recognition of PAMs by PRRs and activation of APC, so we will have co-stimulation. When the cell is normal, there is no inflammation. When there is no inflammation, there is no co-stimulation. Okay? Another mechanism is inhibition or suppression by inhibitory receptors. They are opposite to co-stimulators or activatory receptors. We have some receptors including CTLA4 and PD1 and many others. They are inhibitory receptors. We have activatory receptors including TCR, BCR, and co-stimulators and co-receptors. All of them are activatory receptors. But those receptors are inhibitory. They are like switch off of the cells. Activatory receptors are switch on of the cells, and CTLA4, PD1, and other inhibitory receptors are switched off of the T cells.
This is the self antigen. This is the
They are not antigen, they are receptors.
Yes, I think this in the self antigen in the self antigen.
Yes, in the self cells. For example, you can see it, yes. When there is no inflammation, sometimes we have expression of self-antigen to the T cell, but we have, for example, do not talk about regulatory T cells here. Sometimes we have APC, but instead of CD28, we have CTLA4 and its ligand over there. Sometimes we have PD1 here and PD1 ligand here. They are inhibitory receptors. I'm not going to the details, but just knowing that we have inhibitory, that's why they are called immune checkpoints. Inhibitory receptors are called immune checkpoints. They prevent the activation of immune response against self-antigens. Okay? And I'll talk about it tomorrow. Some tumors use these checkpoints in order to switch off the immune system. They want to escape from immune system. No question? Another mechanism is suppression by regulatory T cells. Regulatory T cells are, I've told you, some suppressory cells that they produce TGF-beta and interleukin-10, two suppressory cytokines, and as a result of production of those cytokines, they can suppress most of the immune cells. So they are suppressory cells. Sometimes instead of activation of, for example, T helper 1 or T helper 2, we will have activation of regulatory cells when there is no inflammation again. And regulatory T cells with different mechanisms will inhibit activation of dendritic cells, T helper cells, B cells, NK cells. They are suppressory cells. How regulatory T cells do this? With different mechanisms. One of these mechanisms is production of suppressory cytokines or anti-inflammatory cytokines. Another mechanism is expression of co-inhibitors or inhibitory receptors. They are like switch off of the T cells. Another mechanism is interleukin-2 depletion. I told you that interleukin-2 is a very important growth factor for clonal expansion. After recognition of the antigen, the first step was clonal expansion. Interleukin-2 is needed for clonal expansion. But regulatory T cells consume all interleukin-2 or most of the interleukin-2 in the environment, and it doesn't allow these cytokines to reach the T helper 1, T helper 2 or other T cells. Another mechanism is again Fas and Fas ligand. I told you regulatory T cells will express Fas ligand. After binding of Fas ligand to the Fas on the normal cells, we will have apoptosis induction in the self-reactive T cell. So a regulatory T cell uses these mechanisms, different mechanisms, in order to prevent the activation of self-reactive T cells, in order to prevent a stimulation of immune system against self-antigen. Okay? Another mechanism is deletion. So, when we have antigen presentation, proper co-stimulation, and production of enough amount of interleukin-2 as a growth factor for clonal expansion, we will have a normal immune response. But sometimes there is no, for example, interleukin-2. Or sometimes there is no co-stimulation. And again, as when we have lack of co-stimulation and growth factors, we mean here interleukin-2. Then the cell will undergo apoptosis, will eliminated by apoptosis. And I talked about Fas and Fas ligand. There is a mechanism in T cells, peripheral tolerance. When a T cell is exposed to self-antigen constantly over time, and it receives several signals by its TCR in the absence of co-stimulation again. So this T cell will express Fas and Fas ligand, and by binding of those Fas and Fas ligand, we will have again activation of apoptosis. Okay, so continuous exposure to any antigen can activate this pathway. Exposure, chronic exposure, or repeated exposure to any antigen can induce this process. And you know that our T cells are continuously exposed to self-antigens. and this continuous exposure to self-antigens induce fast and fast ligand expression on the T cell, and then we will have apoptosis. I told you that this fast and fast ligand, or this fast, is a DES receptor. We have DES receptor, co-stimulator, inhibitory receptors, co-receptors, receptors, they are different from each other. For example, TCR is antigen receptor, CD28, which bind to B7, is a co-stimulator, CD4 is co-receptor, FAS is a DES receptor, CTLA4 is an inhibitory receptor. There are different receptors that they are involved in different aspects of T cell biology or T cell function. Question? Let me skip this slide. That's enough for T cell tolerance. No question about T cell tolerance. We have same mechanisms for B cell tolerance induction. We have again for B cell central tolerance and peripheral tolerance. And but for central tolerance of B cell, if we have a self-reactive B cell after recognition of self-antigens and induction of a strong recognition, the T cell in the first step won't be eliminated. The immune system will give another chance for the self-reactive B cell to change its receptor, which is called receptor editing. What does that mean? It means that in the bone marrow, in developing B cells that are checking against self-antigen, if there is a B cell, for example, I'm a B cell, and if I have a self-reactive receptor in the bone marrow, for B cell, another chance is given to change its receptor. So I have another chance to go and change my receptor. And if after changing my receptors, I cannot recognize self-antigens, it means that this B cell is not self-reactive. But after editing of the receptor, again, if this B cell can recognize that the self-reactive, the self-antigens, it will be eliminated. The receptor editing, we have the receptor editing just for B cell, not T cell. Okay? Yeah or not? Do you know what is receptor editing? The name shows the addition of the receptor, changing of the receptor. And if B cell have a receptor that cannot recognize the antigens in a high affinity, it will be energy again. Let me skip, it doesn't matter. And in the periphery again, we have different mechanisms. The process is same as TCR. We have process of deletion, energy, and regulation by inhibitory receptors for B cell. All of them, we have same mechanisms for the T cell. So I won't explain them again. I'll talk about this slide tomorrow. And we do not have to talk about reproductive immunology. It's a complete course. So, autoimmunity. I'll try to explain autoimmunity as short as possible. I won't explain it in details. I'll try to summarize the content. It is estimated that 5 to 10% of the population will develop in developed countries will have autoimmunity. And these days, autoimmune diseases, the prevalence and incidence of autoimmune diseases increase in developed countries and even in developing countries. So they are, and it is increasing the rate of them. It's very important. Some autoimmune diseases are organ specific and some of them are systemic. Organ specific means that What does autoimmunity mean? Give me a simple definition for autoimmunity. What does autoimmunity mean, please? Anyone else? Anyone else? A simple definition for autoimmunity.
Our immune system attack our cells,
Self-antigens. Yes?
Which means the tolerance process is not work.
Yes. When the tolerance mechanisms don't work, we will have autoimmunity, and the consequence of failure of tolerance mechanisms would be autoimmunity or activation of T cells against self-antigens. Okay? So, in all autoimmune diseases, we have some T cells or B cells that are recognizing self-antigens or inducing immune response against self-antigens. If the target antigens of autoimmunity is expressed only in one organ, it is called organ-specific autoimmune disease. But if the antigen is expressed in multiple organs, it is called systemic autoimmunity. So the definition of organ specificity and systemic immune response is related to the expression of antigen. Okay, if the antigen is in one organ, organ specific. If it is expressed in multi-organ or many organs, it is called systemic autoimmune disease. For example, in lupus, SLE, systemic lupus erythematosus, we have immune complexes in most parts of the body. We have the position of immune complex in most capillaries of the body, and autoimmunity against we have autoimmunity in most organs of the body. But for example, in multiple sclerosis or diabetes type 1, we have just antibody or T cell against pancreas cells, or here we have against neural cells or the myelin of the neurons. Yes, it's kind of autoimmunity in rheumatic fever, not rheumatic arthritis. It's different. In rheumatic fever, I'll talk about it. We have activation of T cells against, B cells, production of antibody against streptococcal antigens, but this antibody can react with cardiac antigens, which is called rheumatic fever. Yes, rheumatic fever is an autoimmune disease, an organ-specific autoimmune disease, because the target antigen is on the cardiac cells or heart cells. Yes. So now I want to talk. Indeed, we have loss of tolerance in autoimmunity. When we have loss of tolerance, we will have induction of autoimmunity. What can induce tolerance failure? Why some people have or tolerance, immunologic tolerance of some people doesn't work properly? What's the reason behind it? Yes, some immune deficiencies is the reason, but not always. Some immune deficiencies could induce autoimmunity, but not always.
Maybe I think there are infection.
Yes, sometimes infection, or many times we can say.
HLA gene or
Yes.
HLA, genes,
yes.
mutation, yes. So, as you said, mutation, HLA, infection, immune deficiency, we can say that we can put the reasons of induction of autoimmunity or failure of tolerance in two groups. Some of the reasons are related to the genetic of the people, and other is related to environmental factors. So both genetic factors and environmental factors are involved in the induction of autoimmunity, or are involved in the loss of self-tolerance. So if we want to talk about genetic susceptibility, so many genes are involved with autoimmunity, mutation in different genes. For example, I've told you that we have regulatory systems for all immune responses. Can you tell me some regulatory systems of immune system? Regulatory T cells, yes. As inhibitory or suppressor cytokines, which some of them are produced by regulatory T cells. What else? Regulators of complement system. Okay, regulators of complement system. And so we have some other regulators that I'm not going to talk about them. Rock. What do you mean by rock? Reactive oxygen species? No, they are the products of neutrophil and macrophage and phagocytic cells. They are not regulators. They are toxic to the microorganism. So, when we talk about regulatory systems, that they are like inhibitory molecules, CTLA4, regulatory T cells, suppressor cytokines, regulator of complements, and inhibitory receptors including CTLA4 and PD-1 and many others. So when these regulators, when a person has mutation in these regulators, that these regulators cannot work properly, so we will have autoimmunity. Genetic susceptibility means, indeed in some part means this, that this regulatory system cannot work properly. Okay? Yes or not? Clear? We have some regulators in immune system. What does regulation mean in immune system? It means we have something that prevents immune activation. Whenever we do not need immune activation, we have something to stop immune activation, to stop immune responses, including inhibitory receptors, regulatory T cells, regulatory suppressor cytokines, or for example, regulators of complement. They prevent the activation of some parts of immune system in order to prevent autoimmunity. But when a person has some mutation in these regulators that they cannot work properly, what will happen? When a regulator of immune system cannot work properly, it means that this regulator cannot stop immune response. So we will have unwanted immune response. Okay, got it? And one important gene again that is involved with autoimmunity and many other immune-related diseases are HLA genes. HLA genes are involved in many autoimmune diseases. I'll show you a table. You might have seen the table in your slides. So there are genetic susceptibilities, HLA and some other genes that I put some tables in the slides. That's I'll talk about them maybe. The other factors are environmental factors. For example, it's very simple. I have a normal tissue and normal protein and normal antigen. When I have a normal protein, normal tissue and normal antigen, I do not have any immune response against these normal antigens. But if I have a mutation in my self-antigens, if I have a mutation and as a result of mutation, the composition of the self-antigen changes, then we will have a new antigen. That new antigen can activate immune system. Okay? So any mutation, any chemical agents in the environment, including air pollution, injury, UV radiation, microorganisms, anything that can change the composition of the self-antigens. Any changes in the composition of self-antigen can induce immune response. What does that mean? It means that, for example, this antigen is a self-antigen and I have tolerance against this self-antigen. But as any reason, we have this part of the table is broken by anything, so there is a change here. And this change is not normal for me. And these changes can activate immune response. Okay? So, and environmental factors can induce change in our self-antigens. And those changes can activate immune response. And in most of the autoimmune diseases, we have involvement of both environmental and genetic factors in the induction of autoimmunity. And it is said that autoimmune diseases, that's why it is said that autoimmune diseases are a multifactorial disease. Multifactorial disease, what does that mean? Many genetic, many genes, and many environmental factors are involved in the induction of the disease. Okay, that's why over time a person, that's why it is said that if a person has genetic susceptibility, it doesn't mean that this person absolutely will induce autoimmune disease. It needs some other inducers, including environmental factors. We need both of them, both factors, both group of the factors. They are not just one factor. For example, if you know that what does apoptosis mean? Programmed cell death. And when we have programmed cell death, several genes are involved in the control of apoptosis and in the induction of apoptosis. Several genes are involved. If a person has a mutation in one apoptosis gene, or let me ask a question in other words, if a person cannot induce apoptosis, what will happen to that person in terms of immune response? The question is this: we have a person that cannot induce apoptosis. In terms of immune response, I mean, yes, the cancer is a different area. In terms of immune response, what? Why? I want the mechanisms. The question is this: we have a person that cannot induce apoptosis. The apoptosis pathway is broken in that person for any reason. What will happen to its immune system? Not for, yes, it's possible to have cancer, it's possible to have some malformation in the fetal-maternal, but I'm talking about immune system. What will happen for immune system?
How can you kill the infected cell without apoptosis?
The first consequence is that cytotoxic T cells kill the target cell by induction of apoptosis. So this is one mechanism. Yes, that's will be.
The immune system not affected because there are many mechanisms for death of the immune systems.
Yes, no. Immune system is affected, of course. Don't you remember from tolerance, most self-reactive T cells were eliminated by apoptosis, both in central tolerance and peripheral tolerance. So when there is no apoptosis, we won't have elimination of self-reactive T cells. Then we will. So some mutations in some proteins of apoptosis can affect autoimmunity, can induce autoimmunity, because we do not have proper elimination of self-reactive T cells and B cells. But other consequences, of course, is possible.
Doctor, in the reality, there is a mutation in the
Yes. That's why it's written here. Or inadequate function of inhibitory receptors. I talked about them. I'm talking about the genes that are involved in autoimmune diseases. Or defective number or function of regulatory T cells. Or defects in negative selection or for T cells and B cells. They are the reasons for the loss of tolerance. Or chronic inflammation. It's very important. I'll talk about it. And any excessive stimulation of the immune response. Do you remember I told you that for all immune responses we have such pattern. We have antigen exposure, activation of immune response, elimination of the antigen by immune system. After elimination, we need contraction in immune response. If this contraction doesn't occur, it is possible to have excessive and unwanted immune response, and the possibility of autoimmunity will increase. So after all immune response, we need contraction, we need decrease in immune response after elimination of the antigen. And I've told you that this contraction, this decrease in immune response is made by regulatory T cells, is made by inhibitory receptors, is made by inhibitory or suppressory cytokines. Any defects in these regulatory systems can lead to an excessive stimulation of immune system. That this excessive stimulation can lead to the expression of co-stimulators and then autoimmunity. You saw that in peripheral tolerance, the most important thing is co-stimulation. Okay? So most autoimmunities are polygenic. It means many genetic factors are involved and even many environmental factors. But these susceptibility genes only increase the risk of autoimmune disease. It doesn't mean that a person will absolutely develop autoimmune disease. What does that mean? It means, let me give you this table, please. Have here, you can see the different HLA alleles that is related to different autoimmune diseases. For example, DRB1 is HLA class 2, one version of HLA class 2, one allele of HLA class 2. That if a person, it means that if a person has this allele, it has higher risk of having rheumatic arthritis. Or if a person has, for example, HLA-B27, it will higher risk to develop ankylosing spondylitis, another autoimmune disease. So we have different alleles of HLA that each one is related to different autoimmune diseases, including diabetes, rheumatoid arthritis, multiple sclerosis, lupus, ankylosing, and many others. It's very important to know that if a person, for example if me, have one of these alleles, it doesn't mean that I will definitely develop autoimmune disease. I just have a higher risk. Because there is need for environmental factors as well. If I'm exposed to other environmental factors in my lifetime, I will develop autoimmunity. But if I do not expose to some environmental factors, including viruses, microorganisms, UV, and many others, it is possible that I won't develop autoimmune disease. And environmental factors, it's multifactorial. So just having one allele is not enough to induce the disease.
There is only genetic, but don't have environmental.
But
there is not
yes.
but high risk.
We have higher risk of induction. If we have exposed to environmental factor, we have in higher risk of induction of autoimmunity. If we receive some mutations from our parents, and if we are exposed to environmental factors, it's possible to induce autoimmunity. let me skip these tables. You are not need to memorize the content of these tables. They are the examples of mutation. For example, you can see here CTLA4, the genes that are involved in with autoimmune diseases. You do not need to memorize them. So, few slides are left. I know you are tired. No, more than 7. Because you came to the class very, very lately after your rest in the beginning of the class, so we can extend the class at least for 1 hour. But I'm not going to do that. Few slides are left. One of the most important factors, environmental factors, that is are important in the induction of autoimmune disease is infections or infectious agents. They are very important in the induction of autoimmunity. So, have a look here. The first mechanism is induction of co-stimulators on the APCs. If you remember, I told you that in normal circumstances, we have a normal cell. Okay? In a normal circumstances, we have a normal cell which is expressing self-antigens on the cleft of MHC, but there is no co-stimulation. If T cell binds to this MHC, we won't have an immune response. We won't have activation of this T cell because there is no co-stimulation between them. The cell is normal, there is no inflammation, there is no inflammatory cytokines, there is no involvement of PRRs, so there is no co-stimulation. But what if this cell become activated, become infected with some, for example, virus? Or if this phagocytic cell phagocytoses normal antigens and it is exposed to the microbe, and this microbe will induce expression of co-stimulators. So the peptide is self-peptide, but co-stimulators is a result of microbe exposure. So now immune system T cells can become activated against self-antigen. Clear? No? It's not clear, it's because you haven't listened to the previous slides. Let me bring the slide. Here, this is an APC. It is presenting self-antigens, but in normal circumstances, there is no co-stimulation. When there is no co-stimulation, we will have energy or unresponsiveness. We won't have any response because there is no co-stimulation. Okay? There is no co-stimulation, so there is no immune response. But if this APC is exposed to infection, for example, E. coli, it will express co-stimulators, including B7. Then we will have activation of the naive T cell against self-antigen. So self-antigen alone cannot activate immune system, but self-antigen in the presence of infectious agents can activate T cell, because in the presence of infection we have co-stimulation. Okay? Or not?
Yes.
Yes? Yes? So the first mechanism by which infectious or infections can induce autoimmunity is induction of co-stimulators on the APC. Okay? Another mechanism is molecular mimicry. Molecular mimicry. It means we have a phenomenon in TCR and BCR which is called cross-reaction. Cross-reaction. Cross-reaction means that if there is similarity between epitopes of one antigen, okay, if there is similarity between the epitopes of one antigen, the antibody or TCR which is produced against the first antigens, as a result of similarity, it can bind to the second antigen as well. Okay? It means cross-reaction. Have a look here. Cross-reaction means we have an antibody that this antibody is against, for example, this antigen. This part of this antigen. This antibody is against this part of this antigen. And if we have another table here that this part of these two tables is similar, this antibody that is against this antigen can bind to the second antigen as well, because they are similar to each other. As a result of similarity between antigens, the antibody that is produced against this one can bind to the other one, because they are similar. We have two antigens that they are similar. So you know, think about the lock and key. We have two locks that one key can open both of them because they are similar. As a result of similarity, one antibody can bind to both of them. Okay? It means cross-reaction. So imagine in rheumatic fever, after a streptococcus infection in the throat of the patient will produce antibody against one protein of the streptococcus, which is called M protein. You've had this test in your practical course. If a patient produces antibody against M protein of streptococcus, this antibody can bind to cardiac or heart antigens of the patient, because in the cardiac or hearts of the patient there is some antigens similar to the M protein of streptococcus. Before entrance of streptococcus, there was tolerance against self-antigens. But after entrance of streptococcus and as a result of immune response and production of antibody, now this antibody can cross-react with heart antigens and induce autoimmunity. It is called molecular mimicry. Clear or not? Clear or not? No? So, for example, we have a microbial antigen here. This is a microbial antigen. And of course, we have immune response normally against this microbial antigen. But in normal tissue, this is a cell tissue, this tissue expresses some antigens, that this antigen is similar to the antigen of the microbe. There is similarity between self-antigen and antigen of the microbe. So this T cell that is reacted against this antigen, now it can bind to self-antigen as well. So the T cell that is activated against foreign antigen, now it can attack self-tissue and self-antigen, because the antigens are similar. Okay? It is called molecular mimicry. So molecular mimicry means that there is similarity between the antigen of microorganisms and self-antigens. And as a result, and production of TCR or antibody against this microbe, we have cross-reaction between this antibody or TCR with self-antigens. Yes. Before entrance of the microbe, we had tolerance. We didn't have any immune response against this self-antigen. But as a result of presence of microbe and induction of inflammation and immune response, then we will have autoreactive T cells. But indeed, this T cell is against non-self antigen. But as a result of similarity between them, it can react with both antigens. Okay, got it or not? Clear? Somehow? Sometimes, so the first role of infection in the induction of autoimmunity is induction of co-stimulators on the APCs. The second one was molecular mimicry. The third one is alteration in the structure of the antigen. I told you, microorganisms can produce enzymes or any other products that this enzyme or products can change the structure of self-protein. Any changes in the self-proteins can induce immune response. I talked about that. Another mechanism is tissue injury and release of the antigens. Some antigens, including antigens in the cornea of the eye or myelin basic protein of the neural system, they are sequestered, they are hidden from immune system. Again, we have some antigens in the body that you can see two examples here. Corneal antigens of the eye, the lens and cornea have some antigens that they are hidden from immune system. It means immune system doesn't have access to antigens of lens and corneal of the eye. Or immune system doesn't have access to a myelin basic protein of the neural system. But sometimes tissue injury as a result of infection can release these antigens. So there is an antigen which is hidden from immune system. Immune system doesn't have access to this antigen. As a result of one infection, injury as a result of infection, we have release of the antigen. So now immune system has access to the antigen. So we will have activation of immune system. So until now, I told you 4 mechanisms by which an infection can induce autoimmunity: induction of co-stimulators, molecular mimicry, alteration or any changes in the structure of the self-antigens, and releasing the hidden or sequestered antigens. Okay? Of course, hormonal factors influence in autoimmunity. That's why most autoimmune diseases are more prevalent in women compared to men. Sometimes 10 times the risk of autoimmunity is higher in women because of hormonal difference in the hormones of the men and women, or female and male. And this table is examples of some diseases. You do not need to memorize all content of this table, but you need to memorize these diseases. This one, I have talked about it. This one, this one, this one. You need to memorize all the contents of these diseases from this table. You can study from the table. And this one, that's it, and this one. You need to study these diseases from the diseases. For example, in autoimmune hemolytic anemia, the target antigen is on the RBC, and autoantibodies we have against the RBC. I've talked about it. That's all. Salam. If there is no question.